Chapter 4, Carbon and the Molecular Diversity of Life. Now, to date, we started off the year learning about the 8 characteristics and what it means to have life. In other words, we talked about Gerstner. Then we moved into an area of how we study that life, and that life had to do with the scientific process called the scientific method. We moved into an area of talking about how life is made up, and life is made up of 70 to 95% water. So we covered the water molecule as being the primary inorganic molecule to life. Now in this next chapter we're going to be talking about the principal organic molecule, which consists of carbon. As a brief overview, again, all cells that are 70 to 95% water, the rest consists mostly of carbon-based compounds. Carbon is unparalleled in its ability to form large, complex, and diverse molecules. Now, hopefully, as we progress with these slides, you folks will remember or hopefully recall that there are large, complex, diverse molecules in our body that make life possible. For instance, proteins, DNA, carbohydrates, lipids, and so forth. But these all contain the element carbon, and they all distinguish living from non-living matter. Now here is a picture that is depicting a great deal of life, and it was believed that that carbon was an element that solely made up life, and it couldn't be synthesized. It couldn't be synthesized in a laboratory setting. That carbon could only be derived from living things. And the definition of organic chemistry, it's the study of compounds that contain the element carbon. These compounds range from very simple, such as carbohydrates, to very large ones, which consist of our large polymers or macromolecules. Most organic compounds contain hydrogen in the addition to the carbon atom. Now I want to talk about two terms. The first term being vitalism. Vitalism is the idea that organic compounds arise only in organisms, was disproved by chemists who actually concluded that that carbons could actually be synthesized in a laboratory setting. So vitalism, again, is the idea that organic compounds can only come from living things. Now, as years progressed and chemists began to study these organic compounds, it was believed that no, mechanism is the view, that all natural phenomena are governed by physical and chemical laws. So what does that mean? It meant that these carbon compounds could indeed be produced in a laboratory setting. Now there is an experiment by a famous chemist by the name of Stanley Miller. Stanley Miller decided that he was going to take some seawater and he was going to put it in a flask-like closed container, and we were going to offer some heat to it. As the solution was heated up, our gases progressed upward into a vapor, into a new chamber that we are considering the atmosphere. Electrodes, some electricity was actually introduced to it, and with that said, you've got some compounds, you've got NH3 and NH2O that condensed down into a collection tube, which was later tested in a chemical analysis run. In conclusion, Stanley Miller suggested that carbon components didn't just come from nature, but they can also be produced in a laboratory setting. Concept 4.2 is carbon atoms can form diverse molecules by bonding to 4 other atoms. Now, how is this done? It's done through something called electron configuration. It's the key to an atom's characteristic. It's the configuration that determines the kinds and numbers of bonds of an atom will form with other atoms. So it's the kinds and the number of bonds an atom will form with other atoms. In consideration of the formation of the bonds, 4 valence electrons. So with 4 valence electrons, carbon can form 4 covalent bonds with a variety of atoms. Remember, it is covalent bonds. This is considered tetravalence because it contains 4 bonds. So this tetravalence makes large complex molecules possible. In molecules with multiple carbons, each carbon bonded to four other atoms has a tetrahedral shape, and we're going to talk about those shapes in a moment. However, when two carbon atoms are joined by a double bond, so when there's a double bond between those central carbon atoms, the molecule will take on a flat shape. This next slide will show that. We've got three different examples. We've got methane, we've got ethane, and we've got ethene. When you've got the carbon that is centrally located, bonded to four different hydrogen, you're taking on a tetrahedral shape. So it looks quite triangular. The ethene has a double carbon that's centrally located with hydrogen surrounding it, and it takes the shape of a tetrahedral, but it's a double tetrahedral. Ethene, however, has carbon that contains a central double bond. And again, when you have that double bond, it is going to take on a flat shape. The electron configuration of carbon gives it covalent compatibility with many different elements. So what does that mean? It says, well, it means that it doesn't just bond with hydrogen, but it can also bind with other atoms, for instance, oxygen and nitrogen. The valences of carbon and its most frequent partners, which are hydrogen, oxygen, and nitrogen, are the building code. So these three are considered the building code that governs the architecture of living molecules. I always like to refer to it as CHON, building code to life. Now, in this slide, we see that it's the valence electrons, which are the electrons in the outermost shell, determines how that particular element is going to bind with another. Now, hydrogen is an exception to the rule, but for oxygen, nitrogen, and carbon, we have to take a look at the empty shells, so the empty spots here, which will determine how that atom will bond. So you want a total of 8 in the outermost shell. And right here we've got 1, 2, 3, 4, 5, 6. It's short 2, so therefore it will look for another element that only has 2 electrons in its outer shell. Nitrogen has 1, 2, 3, 4, 5. It is shy 3, so therefore nitrogen will look for something that another atom that has 3 electrons in its outermost shell to bring it to 8, and so forth. With carbon, it is shy 1, 2, 3, 4. It will look for something that has 4 to bring that to 8. Carbon atoms can partner with atoms other than hydrogen. For example, carbon dioxide, urea, and here we see the molecular structure for urea. We've got carbon, the central carbon, double bonded to the oxygen, and you've got NH2 on this side, and you have NH2 on this side, which is considered an amino group. Molecular diversity arising from carbon skeleton variation. Now carbons, the way the carbons are arranged, can vary. They can vary in length and they can vary in shape. For instance, we've got something called a chain of carbon atoms where you've got the carbons that are centrally located forming chains. Those chains can have single bonds or they can have double bonds. But the bottom line is that they are in chains. Carbons can also branch. So here we've got the chain, but all of a sudden now your carbon tends to branch. And actually, the way that the carbons are situated determines the compound that it is. This particular one is butane, and this one is methylpropane. Now the carbons can also form rings. For instance, in cyclohexane, which by the way, just like in any language, in chemical language, this particular compound here can be abbreviated, showing a hexagon. Now we also have a ring with double bonds, and that would be the abbreviation of a benzene. So again, we've got lengths and we've got shapes. We've got our chains, we've got our branching, and we've got our rings. There are also something called hydrocarbons. These are organic molecules consisting of only carbon and hydrogen, so they're only carbon and hydrogen. Many organic molecules, such as fats, have hydrocarbon compounds. Now, they are rare within the human body, but they are a component of such things such as fats. Hydrocarbons can undergo reactions that release a large amount of energy. Hydrocarbons are actually used in petroleum, and petroleum is used for the production of energy. In this slide, we've got a representation here of some adipose cells. Adipose are fat tissue, and as you can see here, we've taken this cell out, we've enlarged it, and here we have our hydrocarbons here. We've got our hydrogen backbone, or I'm sorry, our carbon backbone with the hydrogens that surround it. In organic chemistry, compounds can form things such as isomers. Isomers are compounds with the same molecular formula but different structures and properties. And when they are arranged differently, whether you've got a shifting of functional groups or you've got a mirrored image, those can actually change what that particular compound is. We have three different examples here. We've got structural isomers, we've got geometric isomers, and we've got enantiomer isomers. The structural isomers have different covalent arrangements of their atoms. So we've got different covalent arrangements. We've got geometric, which has the same covalent arrangements, but different in spatial arrangements, so how they are organized. And we've got the enantiomers, which are isomers that are mirrored image of each other. It's like putting two hands together, and your hands are mirrored images of each other. So here we have a structural isomer. So we have the same number of carbons, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, but they're arranged differently. And when they're arranged differently, we've got pentane here, and we've got 2-methylbutane here. So the arrangement will determine which particular compound we are talking about. So in something called a geometric isomer, remember we've got the same arrangement of the backbone, but take a look. We've got our functional groups, which are located in different spots here. And actually, when the two functional groups are on the same side, it's considered a cis isomer. When they are on opposite sides, they are called a trans isomer. And with the enantiomer, we've got a mirrored image. And again, the example would be the hands being put on each other, and they are mirrored images of each other. And here we have them in a larger view. They're also given the letter L and D in an enantiomer depending on where the functional group is located. Now, the enantiomers are important in the pharmaceutical industry. Two enantiomers of a drug may have different effects. Differing effects of enantiomers demonstrate that organisms are sensitive to even subtle variations in molecules. For instance, we've got two drugs here. We've got the ibuprofen and we've got albuterol. Ibuprofen is used as an inflammatory pharmaceutical drug, and the albuterol is used in asthma. If you have a different arrangement of that functional group, you could end up with two very distinct drugs. For instance, in this particular case, you've got ibuprofen and R ibuprofen and R albuterol and S albuterol. So again, we've got the same number of carbons, the same number of hydrogens and oxygens, but in a different arrangement, causing us the end result to have two distinct compounds. A small number of chemical groups are key to the functioning of biological molecules. Distinctive properties of organic molecules depend not only on the carbon skeleton, but also on the molecular the components attached to it. So in other words, the functional groups. A number of characteristic groups are often attached to skeletons of organic molecules. So these functional groups are the components of organic molecules that are most commonly involved in chemical reactions. The number and arrangement of functional groups give each molecule its unique properties. So I'm going to underline functional groups to give it its unique properties. Functional group here, functional group here, functional group there. So these two molecules look essentially the same. We've got CH3 here, CH3 there, OH there, OH there. But off onto this ring here, we have a hydrogen and oxygen on this end, and we have a double bonded oxygen here. It gives it, so this functional group will give that particular compound its unique characteristic, for instance, estridyl and testosterone. The 7 functional groups that are most important in the chemistry of life are the following. We've got hydroxyl group, carbonyl group, a carboxyl group, an amino group, a sulfhydryl group, a phosphate group, and a methyl group. Now, I recall back in Living Environment, when you folks took that course a couple years ago, you have definitely heard about some of these. For instance, the carboxyl group is found on the end of a fatty acid. An amino group is found on the end of an amino acid. And I know that those have been common compounds that were discussed in Living Environment. Hydroxyl has an OH as a functional group. Carbonyl has a carbon double bonded to an oxygen. Carboxyl has a COOH with one of the oxygens, with the sole oxygen being double bonded to the carbon. We've got the amino group, which is the NH2. Sulfhydryl, which is SH, sometimes written but rarely HS. We've got a phosphate group, which is PO4. And we got the methyl group, which is CH3. And the functional groups give that particular compound its characteristics. And I'm going to move through these slides. You can stop these slides if you wish, but at this point we're going to move right through them. Each one of these slides is given the structure, it's given the example, and it's giving some functional properties and some background information about each one of these. ATP. ATP is an important source of energy for cellular process. Now, you covered the whole idea of respiration and living environment when we talked about the human body. One phosphate molecule, which is called the adenosine triphosphate, ATP, is the primary energy transferring molecule in the cell. It's very important in AP Bio. ATP consists of an organic molecule called the adenosine, which is attached to a string of 3 phosphate groups. Here we see the carboxyl group. Now what's different here, the arrangement looks very similar, but our functional group here is different, given that compound its own characteristic. The chemical elements of life, a review. The versatility of carbon makes possible the great diversity of organic molecules. Variation at the molecular level lies at the foundation of all biological diversity. Here we see the making of ATP. Actually, this is the degradation of ATP being broken down into ADP plus phosphate. And the next several slides are just showing you that there is versatility in organic compounds. Some Lewis dot diagrams, abbreviated version of how an atom will bond with another atom. Don't let these structures frighten you off. We'll be doing some practice, and I'm sure you'll master it. So at the end of this discussion, here are some important points that you should be able to take home with you. Explain how carbon's electron configuration explains its ability to form large, complex, diverse organic molecules. And as the week progresses, we are going to be talking about different divisions of these complex organic molecules. You should be able to describe how carbon skeletons may vary and explain how this variation contributes to the diversity and complexity of organic molecules. And you should also be able to distinguish among the three types of isomers: the structural, the geometric, and the enantiomer. This is critical that you should be able to name the functional groups found in organic molecules, describe the basic structure of each, and outline the chemical properties of the organic molecules in which they occur. When watching this on your own, you can go back in the previous slides and stop it and look for, when we broke it down into the different functional groups, it tells the key points about each one of those functional groups. You should also be able to explain how ATP functions as the primary energy transfer molecule in living cells. Over the next couple days, we're going to be talking about that energy. That concludes our discussion.